Researchers are growing parts of the human brain in petri dishes to study and develop treatments for disorders like autism and schizophrenia. It's part of a movement to produce organoids, which are miniature versions of body's organs for medical research. These tiny balls of tissue are opening a new window into the human brain. They're clumps of living cells that make up the cerebral cortex, which plays a key role in memory, thought, and language. The cells are being grown right here in Dr. Sergio Pasca's laboratory at Stanford University. Until now, Pasca could not directly study key portions of a living brain. These whitish balls of human brain tissue, smaller than a pea, are created from human skin cells, including some from people with autism. Each one carries the DNA of the person it came from.
I think this is exciting because, it's essentially providing us another tool in the arsenal of tools that we have for studying the brain.
This laboratory isn't the only one to grow its own cell structures. Dozens of labs are growing lumps of human brain tissue for study, a practice that drew notice in 2013 when researchers said they had created mini brains that contained multiple major parts of the fetal organ.
What we're trying to do is we're trying to make very specific parts of the brain that are important for very specific diseases and study those in isolation in a dish. We're not trying to make an entire brain. That is not really the purpose.
Although brain cells in lab-grown tissues show some activity, nobody has created fully functioning adult human brains. There have been reports of these in scientific journals, but these versions mimic only one or more parts of the fetal brain. There's an unproven claim this year that a lab had created a nearly complete fetal brain, but this has not been backed by a medical journal. This research aims to test possible treatments and perhaps move toward supplying replacements for transplants. Scientists as of now have made organoids representing the intestine, prostate, kidney, thyroid, retina, and liver. According to Arnold Kriegstein at UCLA or University of California, Los Angeles, this is enabling scientists to research with human tissues rather than substitute animal tissues.
Those of us studying nervous system disorders had to model them in animals. But now we can actually model them using human cells from patients who actually have the actual disease. So I think it gets us much closer to treating human diseases than we ever were before. And that's not only true in the nervous system, it's true in organoids from the liver and from the pancreas and even from the heart. So the entire field of medicine, I think, is going to be impacted by advances in the stem cell biology arena. These are remarkable models for studying human diseases and will hopefully open up new targets for therapy and create a better understanding of some of these profound disorders.
But these are far from functioning brains. Although these are clumps of cells in a 3D structure, the different regions of these organoids don't communicate with each other the way our brains are able to. Also, there's no blood supply, which limits their growth and keeps them in a primitive state. Because of this, the research doesn't yet raise ethical debate, according to Hank Greeley, who is a specialist in law and bioscientist at Stanford.
Tiny little bits of brain, I think, don't raise any serious questions. A little brain organoid is not going to be sitting back thinking, who am I, where am I, why am I here?
But even Greeley admits that nothing will stay the same.
And yet, I think it's a good time for us to think about what limits, if any, we'd want to put on this kind of research.